This video is going to get you through the senses notes in lecture. This is something that we've already talked about in the lab, and so a lot of this is going to seem like it's repeat information, so I'm also going to go maybe a little bit faster than I normally do through here, except for stuff that I didn't already cover in the lab. So to start out, we talk about sensation versus perception. Sensation is a detection of a stimulus from the outside world. For example, in this picture, you guys can all sense the little black spots that exist in this picture. If the sensation is strong enough, you can turn sensation into perception, which is where you actually start to integrate that information and turn it into something that's meaningful. For example, some people can perceive a horse and rider in this picture, like here's the horse head, here's the rider, here's the horse tail. Some people don't though. So perception has to do with sort of background information and how you integrate all of that information. And so perception is different from person to person. It's sort of like, it's like art, essentially. Everybody can sense the same thing when they're looking at that painting, but they don't perceive the same thing because you integrate that information differently. Like, I've always hated Jackson Pollock paintings because I can throw paint at a wall and end up with the same sort of thing. But some people absolutely love Jackson Pollock paintings, and so that perception is just a little bit different there. A sensory receptor is a cell or an organ in the body that can detect a particular type of sense or stimulus. The next several things that you have in the notes that all end in receptor, those are specific types of sensory receptors. They are named based on what they can detect. So a mechanoreceptor can detect mechanical stimuli. Mechanical means like pulling or stretching or bending, anything that has a mechanical or physical force, a mechanoreceptor is going to be able to detect that. Your sense of touch is very reliant on mechanoreceptors, but so is your sense of hearing, and so it doesn't necessarily always correspond to what you think it's going to be. Thermoreceptors. Thermo means temperature, so thermoreceptors detect temperature changes. That can help you know whether or not you're touching a hot stove, but it can also be used to detect internal stimuli so that your brain knows what your body temperature is right now so that you can help to regulate that. Pain receptors do exactly what their name says. Those are also called nociceptors. They detect damaging stimuli that can actually hurt your tissues. They're really just a special kind of mechanoreceptor, but whatever, we'll go ahead and put them as their own separate thing. Proprioceptors detect the position of your body. Like, proprioceptors know whether your knee is bent or straight right now. They know whether your spine is flexed or extended right now. They just know the position of your body, and proprioception is very important for movement as a whole because you can't move gracefully if you don't know where your body is currently. If you've ever watched a pubescent boy go through a growth spurt, their proprioceptors don't really keep up with their growth, and so they get really clumsy and they'll do stuff like fall on their face and run into door frames, and it's because their proprioception is broken while they're doing that growth spurt, but it usually hooks back up later on and they'll get it back. Photoreceptors detect light. Chemoreceptors detect chemicals. Your sense of smell and taste is very reliant on chemoreceptors, and that's what this picture is actually trying to show you. So in this case, we have a sugar molecule like glucose. These are all of the chemoreceptors. When they detect that chemical, they're going to send off an impulse to the brain, and then the brain is going to be aware that that sugar is actually present in there. All right, after that, let's see if I have a slide for transduction. Transduction is the ability to convert one type of energy to another type of energy. So your brain works off of chemical signals and electrical signals, but you have these different receptors that all can sense different types of stimuli. So in this case, we have a mechanoreceptor. This particular type of mechanoreceptor is called a hair cell because it has these little hairs on one side. If these hairs bend, this cell fires off an impulse, and then it's going to tell the brain. So this cell is capable of transduction because it can convert mechanical energy into chemical and then electrical energy that the brain is going to actually be able to detect. All of your receptors have to be capable of transduction, otherwise you can't sense that stimulus. And so transduction is critically important for all of this to work. Sensory adaptation, I'm not sure if I have a slide that goes along with that, so I'll just show you this wonderful looking snake right here. Sensory adaptation is you getting used to a stimulus and basically you start to ignore it. Like probably at some point in your life, you have been forced to sit next to a person who smelled like they hadn't showered in 3 weeks. They had some fairly nasty body odor. If you sat next to them for an entire hour and a half class, you wouldn't notice that smell after a certain point. That is sensory adaptation. Another example of that, a lot of people in our area live next to train tracks, and when you first move next to the train track, you hear every single train that drives by, but after a while it starts to just become background noise and you don't hear it anymore. And so that's another example of sensory adaptation. It's getting used to a stimulus because it's ever present, essentially. What are olfaction and gustation? So olfaction is your sense of smell. Gustation is your sense of taste, and both of those rely on chemoreceptors to detect specific chemical shapes that your brain can then perceive as like a sugary taste or a floral smell or whatever it happens to be. After that, I gave you this picture to label. This is again something we're already doing in the lab. So since I've already given you a lab video that corresponds to all of this, just pause the video and get what you need. I'm not going to harp on it too much again. What is the pathway of light through the eye? So I'll do that in the picture. So we're going to first pass through the cornea, then through aqueous humor, through the pupil, to the lens, through vitreous humor, to the retina, and then will be absorbed by the choroid. Okay, after that, I guess I'll have to stay on this picture. I haven't updated this presentation in a long time. What's the function of the following? Starting with the sclera. The sclera protects the eye and maintains the shape. The cornea lets light into the eye. Both of those, the sclera and the cornea, they are avascular. There are no blood vessels in those two layers. The choroid is the vascular layer of the eye. That means it has the blood vessels and it supplies nutrients and respiratory gases and things like that. But it's also a very dark layer that's going to absorb light and prevent the light from bouncing around in your eye and making things appear more blurry. And so the choroid does also play a functionality when it comes to the actual image that's going to be formed. So it absorbs light so it doesn't bounce around. The iris is the colored part of your eye and it controls the size and shape of the pupil. The pupil is the opening that allows light through the vascular layer to hit the lens. The lens is the structure that can focus light specifically on the fovea centralis, which is the center focal point of vision where most of the cones in your eye are going to be found. So the lens's job is to focus the light right there. The retina is this whole yellow layer that contains the rods and cones. All of the rods and cones, the axons, merge to form the optic nerve, which is going to take visual information back to the brain. The rods can be found all over the retina. They are very sensitive to light, but they can't see color. But they do help you see, like if you wake up in the middle of the night and you try to go to the bathroom, but you don't want to turn a light on, they'll help you navigate to the bathroom so you can still see the shapes of things, like there's a dresser over here, don't stub your toe sort of thing. Most of the cones of your eye are found in the fovea. Cones are not very sensitive to light, which means they need bright light in order to work, but they can differentiate between different colors of light. We're not really going to talk about compound eyes all that much, but compound eyes like this fly has right here, what that means is each one of these structures has its own lens and focuses its light onto its own specific little place, and so they just have different vision than we do, essentially. This picture tries to show you what you have to do in order to see things that are up close. I mentioned in lab that you have to carry out the process of accommodation in order to see things that are up close. What that means is you're going to sort of, I don't know even how to describe it, you're going to make your lens fatter so that you can focus the light better when you're seeing something up close. The natural shape of your lens is a little bit more stretched out. Assuming that you have 20/20 vision. Again, if you're myopic or hyperopic, this is all different, but this is what your lens is supposed to do, is make sure no matter where the object is that you're trying to see, we always have to focus the light onto that fovea so that you can see it really clearly. since we're not counting or working together in class, I'm not going to worry about this little part right here. These couple of slides that I have coming up show you what happens when your eye doesn't focus your light correctly. So in the case of this picture, the lens is trying to focus the light onto the fovea, but because the lens is either broken or your eyeball maybe has the wrong shape, the light is being focused in front of the fovea. So when it hits the fovea, it's actually spread out a little bit more. So what they would do if this is happening to your eye is they would make you specific lenses that are going to help to focus the light better. This is how all eyeglasses work. They work with your eye to make sure that we're getting things focused correctly. And at least according to that YouTube pop-up, I have a video that corresponds to this. So let's see what the video is. It might be gross. It might be a cataract video, but I don't know. I like gross videos. They make me happy. Yeah. All right. So one of the things that I do discuss in the lab is cataracts, which is a clouding of the lens. I mentioned that you should probably go online and try to find one of these. So here is just one of those. If you don't want to watch this video, just fast forward through this. But I think it's really cool to watch how we can take a person who couldn't see, and at the end of this procedure, they're seeing 20/20 vision out of this eye. It's amazing what we're able to do.
Cataract surgery, Dr. Kevin Smith, Ottawa, Canada. Here we have the patient's eye staring at a bright light. The eye has been frozen with jelly. Initial incisions are made into the clear, dome-shaped cornea.
Mute that. Notice there's no bleeding as we've done that, and that's again because the cornea is avascular. There's no blood vessels in it, so it can't bleed when you stab through it. Now the little bit of bleeding that you see up over here, there's a thin film on top of the eye called the conjunctiva. If you've ever had pink eye, that's infection of the conjunctiva, but that's what's bleeding right there. But most of this whole procedure, we're not going to bleed. Now he's starting to peel back the covering on the lens, and I'm going to unmute him now.
Candy. We are making an opening in the outer candy layer and will be removing the chocolate. The rest of the outer membrane layer will remain to hold the new lens in place. Fluid is used to separate the lens from its outer membrane. You can see the fluid wave in action. We then rotate and free the lens.
That rotation helped to break all the suspensory ligaments that were connecting the lens to the ciliary body. Now this little device is an ultrasound. It uses sound waves to break up the lens better, and it's also a vacuum, so it can suck up the lens as it's breaking it up. And so, yeah, you can notice it's creating little grooves in there, and it's sucking up that bad lens to get rid of it. This part takes a while, so I'm going to kind of skip past some of this. Let's go to right here. So this is just a more sort of high-powered vacuum. It's getting the rest of the membrane out of there, because if it were left in there, it could set up a bacterial infection, so we're getting rid of that. And then, poof, we just put the new lens in. So that's a cataract surgery. That person can now see when they could not before. And again, it's just amazing what we're able to do with technology these days. All right. Don't go back. No. Oh man. This is why I shouldn't just click on things like that. Bink. Okay. I need to be far away from that. Okay. So this is what happens when the eye sees in the other direction. Now the lens is trying to focus the light behind the fovea, so it's blurry when it's hitting the fovea. We need different kinds of corrective lenses for that, essentially. So the rods and cones that I mentioned earlier, they are named based on the shape of the cell. So rods are rod-shaped, cones are cone-shaped. Not only do they have different shapes to the cells, they hook up differently to different neurons. And for that, I wanted to show you this picture. So rods. Multiple rods all talk to a particular cell, which is why rods see things sort of as a fuzzy vision. Cones, on the other hand, each cone connects up to its own specific cell, which is why they're able to give you more crisp color vision. It's because they have a 1-to-1 ratio of the cells that they're hooking up to, although this picture really doesn't show that as well as I wanted it to. We already watched that, so we're good. Next up, I gave you this picture to label. We're also going to add some labels that aren't on here. Again, we've already really done this in the lab, but just to sort of hit the highlights. The external ear consists of the auricle, which is this whole structure. It does have different parts to it, but we're just going to call it all collectively the auricle. This is the external auditory canal, because there is an internal one, believe it or not, but it's usually just called the eustachian tube, which you can see going on right here. The eardrum, let's not call it that. Let's call it by its real name, which is the tympanic membrane. That's what you had to call it in lab, so we might as well call it the same thing here. The middle ear starts at the eardrum, and it consists of 3 ossicles, which are the ear bones. We have the malleus, which is the first one, directly connected to the eardrum. We have the incus, which is the middle bone, and then we have the stapes, which connects to the oval window. Oh my goodness, you can't read any of those, but hopefully you at least remember sort of how to spell them from the lab. Then we get to the inner ear. So the oval window, which is right here, is what separates the middle ear from the inner ear. We have this snail shell looking thing, which is the cochlea. We have these little tubes that are called semicircular canals. And then the middle chunk that connects those two together, that's the vestibule. Special things about the ear. So this should be filled with air. You do have some glands in there that secrete cerumen, which is earwax. If the earwax builds up too much, it can impact in there and then lead to hearing loss, not to mention just uncomfortableness and even sometimes infection in the outer ear. But you shouldn't stick Q-tips in there. If you ever talk to an ear, nose, and throat doctor, they will tell you to never stick anything smaller than your elbow into your ear, which is just their goofy way of saying don't use Q-tips in there. In fact, if you read a box of Q-tips, they specifically tell you not for use in the ear. And that's because if you stick a Q-tip in here, the tympanic membrane is so thin and it is so delicate that it's really easy for you to poke a hole through it, which is not only excruciatingly painful, it will impact your hearing as well. And so sticking Q-tips in there makes you more likely to rupture your eardrum, so don't do it. If you need to clean out your ears, they make stuff, a brand name is called Debrox. You can put that in there and it'll help loosen up any earwax to help clean your ears. But yeah, don't stick Q-tips in there. Your middle ear, this whole section right here, it is also filled with air. We have this eustachian tube that connects back down to the throat so that you can equalize pressure between your middle ear and your outer ear. If you don't have equal pressure, your eardrum can either start to bulge outwards, which makes it more likely to rupture, and it's very painful, or it can start to suck inwards if you create a negative pressure area in there, and that's also fairly painful. So this tube helps to equalize that pressure. Anytime you pop your ears, you're actually letting air pass in one direction or another through this tube so that you can equalize that pressure. The inner ear is filled with fluid, though. That fluid is going to help you actually hear the sounds because it is going to oscillate back and forth and vibrate. So that's what's going to allow you to do the hearing part. All right. What's the function of the following? The eardrum. It vibrates in response to sound, and it amplifies the vibration, which means it makes the sound kind of louder in your ears. That's what your eardrum is supposed to do. Your cochlea contains hair receptors for hearing. So the cochlea is the part of the ear that actually does the hearing. The hair cells can be found within a structure in the cochlea called the organ of Corti. This area right here is the organ of Corti. We've blown it up. This is a hair cell. This is a hair cell. That is a hair cell. The hair cells respond to sound by the bending. The hairs will bend when the sound wave presses on them. And then when the hairs bend, we're going to fire off an impulse and then tell the brain, hey, we bent our hair cells. We heard a specific kind of sound. So again, the hair cells are really what do the hearing. They're in the organ of Corti, which is in the cochlea. The vestibule, and I'm going to come back, I'll do it on this picture. This is the middle chunk of the ear, basically right through here. It's in the inner ear. The vestibule is responsible for static equilibrium. What that means is it tells your brain the position of your head right now. Like, are you standing up? Are you lying on your right side? Are you lying on your back? Your vestibule knows where the position of your head actually is. The semicircular canals, which there's actually 3, you can only see 2 in this picture, they are responsible for what's known as dynamic equilibrium, which is a directional movement. Are you turning to the right? Are you turning to the left? Are you doing a cartwheel? But it's basically which direction are you spinning? That's what your semicircular canals do for you. Let's see, this one sort of gives you your pathway through the ear, so if you wanted that, voila, you got it. What I kind of just want to point out is, notice how the job of the middle ear is to amplify the sound waves. So sound travels as a wave. The amplitude of a wave is from one peak to one trough. The taller the wave is, the louder the sound is. And notice we get a lot taller in the middle ear. So the middle ear's job is to amplify that sound. The eardrum starts it, but the malleus, the incus, and the stapes actually kind of finish that process out. The sound wave is going to start to get shorter as we go through the cochlea as well. Then, just as a different sort of aside, waves can also be described by wavelength. It goes from one peak to the next peak. Different wavelengths are different pitches. Think like different notes, like a B or an A or a G or whatever. I don't remember too much of my music stuff from back in high school, but those are just different notes. And the cochlea can differentiate between different wavelengths depending on which hair cells are actually vibrating in response to the sound. The higher pitched sounds are heard by the hair cells closer to the beginning of the cochlea versus the low pitched sounds, the lower notes, those are heard by the cells way towards the end of the cochlea. All right. This is just my reminder picture about your eardrum. So we're looking through what's known as an otoscope, which is that little lighty thing that a doctor will put up to your ear and look in your ear at. And we're looking at the eardrum. This person has a perforated eardrum, and we've got a lot of inflammation. We should not be this red inside of the eardrum. They more than likely had a middle ear infection that didn't get treated or something, and as a result, they've popped the eardrum. If you ever have children of your own, and you notice that your kids are pulling on their ears a lot or rubbing on their ears, that tends to mean that they have an ear infection and you should take them in to be seen, because it really sucks to pop an eardrum and it's always better to go have a doctor check you out. Now here's the flip side to this. A lot of people tend to think that they need to clean their ears out every single day, and they do the Q-tip thing even though they've been told not to use Q-tips in their ear. Well, cerumen, that earwax that I was talking about earlier, it's actually been shown to have an insect repelling property to it. Not to mention, it's also antibacterial. If you keep your ear too clean, bugs are more likely to actually go into your ears. Some of the more common things that crawl into people's ears, this one happens to be a roach, which, yes, super cringe, but moths like to get into ears as well. And I have not had a bug in my ear personally, but I can imagine the sound of, let's say, a moth fluttering around in your ear, trying really desperately to get out of your ear, but it can't figure out the way out anymore, and how maddening that would be to you, because you're hearing all of this sound and you know there's a bug in your ear and it's driving you crazy. So you would want to like shove anything in there to kill the bug to make the sound stop. But again, if you do that, you're much more likely to damage your tympanic membrane. So ERs deal with bugs in the ears all the time. If you need to go, please make sure you go so that you don't damage the ear out. One of the things that you can do to stop the sounds from happening are, if you have mineral oil, put some mineral oil in your ear. That will at least drown the bug so it stops moving. Then that'll give you time to go to the hospital so that they can get the bug out a little bit later on. But try really hard not to damage your ear. Your ear doesn't repair itself as well as you might think that it does, especially if you rupture the eardrum. So don't over clean your ear, don't stick things in your ear, and if you think you have a bug in there, go to the ER and don't just shove things in your ear. Do we have a thing about motion sickness? We sure do. Okay, so additional information. First up, you had, what is cataracts? Cataracts is clouding of the lens. I've shown you a video now for how we treat that. What is glaucoma? We talked about that in lab. It's a buildup of aqueous humor, which is the fluid in the anterior segment of the eye. We test for it with the puff test. I talked about it in lab. Go to the lab to get more info on that if you want it. How do loud sounds damage the ears? I talked about that in the lab video, so you should be good there. And what causes motion sickness? I talked about that in the lab, and so go get all that information from there. From there, I've given you guys a quiz at the back of the notes. We're going to play this sort of like we did the last one. Complete this quiz, scan it, and then upload it into Blackboard, and then you're done with special senses.